afternoon, all of you. I, Dr. Vivek Srivastav. I thank you for joining us today for the webinar on novel strategies in drug discovery. Honorable Dr. M.N. Patel, sir, Provost, Parul University. Respected Dr. Abhay Dharamsi, sir, Dean, Faculty of Pharmacy and Principal, Parul Institute of Pharmacy. Dr. G. Chakraborty, Principal, Parul Institute of Pharmacy and Research, and Dr. Lalit Lata Jha, Madam, Principal, School of Pharmacy. I welcome Mr. Saurabh Maru, Assistant Professor, NMI-MS University, Sherpur. So, if you are interested in gaining expertise in the field of drug discovery, then this is the webinar for you. And today, we are very lucky to have with us Mr. Saurabh Maru. Mr. Saurabh Maru is an avid researcher and academician, currently working on nutrition and development of exosome-based diagnostic tools. He worked at various peer institutes of India, including NICPR, NCR, Indian Institute of Science, Bangalore, CBST, which is situated at VIT University and which is jointly established by BST India and UTC France. Currently, he is working at NMIMS University, Sherpur campus. He is also serving as associate faculty of IIT Bombay. He has 17 publications to his credit, and he has been invited as a reviewer and associate editor by various publication houses like Oxford University Press, elsewhere BMC and Bentham Science. He served as resource person and in an advisory role in MP Council of Science and Technology Bhopal, Niti Aayog New Delhi, MDRU, Raja Ramanna Center for Advanced Technology, Department of Atomic Energy, Government of India, Niti Aayog MP Regional Outreach Bureau, and Ministry of Information and Broadcasting, Government of India. He was scientific advisor to Research and Extension Wing, Forest Department, MP Government. He also served as expert in Committee of MP COST and Rural Technology Action Group of PSA to Prime Minister, Government of India, to advise on healthcare and rural development in MP. He served as expert at Animal Welfare Board of India, Ministry of Environment and Forest, Climate Change, Government of India during the tenure 2014 to 16. He is member Board of Studies, Department of Pharmacy, SGS ITS Indore, and recently he has signed MOU with Abo Academic University, Finland for research work. He has received many awards in recognition of his scientific and social endeavors from reputed organizations like IUPHAR USA, NITIE Mumbai, Department of Atomic Energy, BRNS IIT Mumbai, SAS Private Limited, and he is founder of social initiative Nirog Facilitator Network working for tribal people of MP, and he has helped more than 4 lakh patients at government hospitals and through government policies. Felicitation. So thank you very much, sir, for coming down to the webinar, and we are really excited to hear from you on this special topic. So it's all over to you, Saurabh sir.
Thank you so much, Vivek sir, for such a interview, such a introduction, and I'm very thankful to Parul University, and I am thankful to the university officials, principal sir, ma'am, and Vivek sir for inviting me for this webinar. Today I'll be discussing about some of the novel strategies which are used in drug discovery process, because in last few years with the advent of artificial intelligence, machine learning, and various insights about the biology, about the molecular markers, and about the technology, we are able to go into much detail into the various aspects of the drug discovery. So when we were having nothing, in a few centuries back, it was really a very difficult task for getting rid from disease. So it was like a situation, if somebody get injured, it becomes very difficult for that person to recover. It's the body itself has to recover by themselves. And that's how the people initially went on. And later on, slowly the things developed into the from the alternative medicine, the various medicinal systems emerged out. And the similar things happens in due course of time with the history. So we are talking usually about the modern medicine, and we found that there is a different parameters in all the medicinal system. So we look forward the specific parameters, and that's how we come across the very specific pills. We call it like magic pills, which was first coined by Paul Ehrlich. So it was long back in 1854 to 1915 time when he told about a magic bullet, and that magic bullet which can selectively affect any physiological function, pathological function, and cures that. In that way, if you take one medicine, that particular pathological problem get wiped. So that was really considered as a magic bullet. And earlier the infections was like, if somebody get infected, then it become very, very difficult to get cured, and many people died because of any sort of infection. So being remain away from or remain unaffected by infection was the way to survive longer. But with that advent of magic bullet and its a concept, it has completely transformed. And you know how these magic bullets are affecting, because they are interacting with various targets, and these targets are proteins. So practically every medicine interact with a certain protein which acts as a target for that particular medicine, and that either inhibit or enhance its a function. So there is around 2 million, more than 2 million targets has been identified in only human body. So you can understand that we have 2 million sites where we can alter the pathology or physiological aspects. So there is a different way how we can alter, how we can find these targets, and this is how the complete transformation of drug discovery process happens. You know, earlier it was mostly evidence-based medicines and evidence-based approaches, like they ate something and they get cured of something. So in that way, they correlated that this can cure that. So likewise, it was not a very empirical way of drug discovery, but now it has completely transformed into different approaches. So the approaches like phenotypic screening, gene association studies, chemoproteomics, transgenic organisms, yeah, imaging biomarkers, and so on. So there is n number of techniques which has evolved out, along with the various instrumentation methods, various chemicals, various advanced and derived type of equipments, which actually made it possible to go into much higher detail of this individual. So here, the most important part which I can consider is the pharmacology, which plays a very important role, because this is the one which translated the laboratory and animal data into the clinical data, and that's how they connected the experimentations with the humans, and this experimentation could be extrapolated. So here the pharmacologist could specifically identify the specific targets, and this target could be taken up by the chemistry people, and could be prepared some molecules how these targets function can be enhanced or subsided. In that way, it finally evolved out to be a drug in the future course of studies. So drug discovery process, I'll just give a fast review over this, that what it is exactly. The first process is target selection and validation for any sort of the disease. So there is a studies of a specific disease mechanisms, and once we understand the disease mechanisms and their specific biomarkers, so based on that, based on the molecular studies, we can find the specific biomarkers, and this requires animal studies. The specific and relevant species need to be selected. There is need to design transgenic animals. The specific conditions has to be induced. Certain agonists and antagonists need to be expressed or need to be understand before going, whether it is enhancing or whether it is inhibiting the particular function, the X drug, and designing of various antibodies, antisense, and RNAi, etc. So these are the various techniques which actually emerged and required for finding the disease mechanism. So we need to understand the various targets based on the mechanism of the disease, the pathophysiology of the disease, and that leads to the discovery process. Once we identify the targets, that could be receptors, ion channels, transporters, enzymes, any signaling molecules, any secondary signaling molecules. And once we are able to modulate that, we can go ahead for some modulation with a specific type of the compounds. And this leads to the search of the lead. So lead search is basically development of the assay or development of a chemical diversity, highly interactive process need to be developed, and in that way we can find that which molecule can affect that specific target, whether it is enhancing or whether it is suppressing, but who is the one which is affecting, and that is called as the lead for that particular disease. And then we need to optimize the lead. Again, we have to go with the in vitro and in vivo in silico studies for the lead optimization to see the selectivity of lead, efficacy in animal models, tolerability, and then the various mechanisms, pharmacokinetics, and various interactive processes to understand whether whatever the compound we come across is really able to work for that particular disease or not, or particular target or not. And then it goes into the developmental stage where we take this drug for animal studies to see the safety of the drug. You know, it's not just the efficacy, but the safety is nowadays prime concern. Whether the drug is doing good or not, but it should not do bad to the people. So it is very important of prime concern. We already seen in pandemic that how various drugs has affected the people. So people has used many drugs like anything, and later on they observe a great number of and a huge type of side effects because of the various drugs used in blindly in this pandemic. So drug candidate studies, and then it goes for the human studies. So once the drug is safe at the preclinical studies, at the animal studies, then it can be extrapolated and used in the humans at the various stages of phase trials, and finally it comes in the approval process. So starting from the position of finding the disease mechanism, and then finding the specific target which is actually involved in the process of disease, and then modulating this, and then once we confirm that this is the only one which is causing the modulation, then we need to study the specific candidates which are affecting that gene. For example, a very simple example, like we come across that prostaglandins are leading to the headache. Okay, so we identify one drug, that is paracetamol, and that is inhibiting prostaglandin. Okay, so it is affecting the COX, and in that way, so the target is COX, lead is paracetamol. So we identify COX versus paracetamol, and we found that it is perfectly fine to inhibit the COX with the paracetamol to inhibit the headache. Okay, so to control the headache. Then we tested this paracetamol in the animals for the safety. Once we found it safe, then we proceed for the human studies. Once it has qualified for the human studies, then we went for drug approval and registration, and then drug, that paracetamol, came into the market maybe in the form of Crocin or Combiflame or like any company who launched this drug. So in preclinical study, the first is target discovery, and there is a various steps of finding the target. Then the lead discovery. So based on this target, once we have identified one target, we test various molecules over this, and that is called as lead discovery. And then once we could found a specific lead, then we can go for the lead optimization. So based on the structure activity data and the ADME data and various other data, safety data, we go for the lead optimization with the help of medicinal chemistry, etc. And then finally, once we found a very specific and the lead which is qualifying our criteria, we go for preclinical studies. So we go for the toxicology and safety study, and then we do the formulation and dose prediction, etc. And then the phase is called as discovery process, and there is around 4.5 lakh compounds came as the lead discovery process. And once it is optimized, you can say that these are the various compounds which finally proceed to the development process. And in each step, there is thousands of such leads get dropped because they are no more effective. So we go with only the effective molecules, starting from the legs of molecules. Then once we qualify all the criteria related to preclinical, that means animal trials, it goes to the human trials. So in human trials, the phase 1 trial is basically for tolerability, that means safety. So we use only the humans who are not having any disease, and we try to look into the safety of the drug, and then further efficacy, and then safety and efficacy. Likewise, there is phase 1, phase 2, and phase 3 trial, and then phase 3 become multi-centric and the multi-ethnic trials. This is the developmental phase of the drug, and you find around 12,000 candidates at this level. So 4.5 lakh to it come to the 12,000 candidate, and finally at the launch, when it qualified for all the requirements of the legal authority, we start launching the drug into the market, and the people will start using it, and there is only less than 1,300 drugs qualifies. So 4.5 lakh to 1,300 is actually the whole life process of the drug which it has to go through. So discovery process requires many things to understand, like there should be an assay to evaluate the activity of compound. So there can be in vitro or in vivo assays like enzyme assay or animal models for pharmacodynamic studies, etc. Then identification of lead compound through the screening or from published literature, and then through the screening of natural products, through the screening of various structure-based designing methods, that is called as rational drug design, and through the compound libraries. So we try to virtually put every molecules with that target so that we can find which is the best fit. And once we are able to develop a proof of concept, then we can go for further studies of the preclinical, like pharmacokinetics, like metabolism, and then off-target activities like the toxicity and safety studies of the drugs. So this is the overall process of the drug discovery. The preclinical to clinical studies requires safety toxicology, that is called as ADME studies, and then its process should be established in the R&D and manufacturing unit. So bioprocessing of the drug, and then pharmaceutical and R&D formulations, and finally it has to pass through all the regulatory bodies so that it can come into the market finally. And then even after the launch of the drug, there is a lot of studies has to be carried out to submit to the regulatory bodies continuously, intermittently, in given time, and that data has to be recorded. Okay, so it is called as like sometimes phase 4 trial, that is the surveillance. So post-marketing surveillance is also performed with the drug candidates. So these are the clinical trials. Phase 1 trials use very few volunteers, 220 to 100 volunteers, and this basically give information about the absorption, metabolism, effect on organs and tissues, and side effects in the because of this. So toxicity is the major concern of the phase 1 trial, and mostly healthy volunteers are employed. In phase 2, the patients are employed, and the effectiveness of the drug is observed. Dose range is also actually understood or recorded. And then in phase 3, 1,000 to hundreds to thousands volunteers are employed, and then benefit versus risk ratio, and any specific side effects or labeling informations are actually worked out. So these are the 3 phases required done for the specific specific informations. In the approval process, the data which we have collected from the preclinical as well as clinical trials need to be submitted to the regulatory bodies, and it is filed as a new drug application. Then there is various regulatory advisory committees and regulatory review team, which actually provides their feedbacks, comments, discussions, and then that has to be finally satisfied for the approval of the drug. So in this way, the drug approval process happens. And when we look into the drug discovery process, there is certain skill sets which is actually required, and that has converged now. Earlier, we were having a specialization in individual, but now we need to be specialized in many specific aspects of the drug discovery. So it is a joint effort, and there is a joint effort of the whole pharmacy professional. And apart from this, there is a very important aspect is the physiological aspects which plays a very vital role in the whole process. So whether it is in vivo, safety, pharmacokinetics, pharmacodynamics, studies, there all the physiology is another important aspect, which is actually very important, apart from the other skill sets. So when we talk about target, the physiology plays an important role to select a specific target, and we need to understand the unmet need of the medical profession so that we can find the specific target required for this. And in that way, we can ultimately lead to the various right processes or right direction for the drug discovery. Okay, so it's not just the animal model, but we need to go ahead with the various advanced technologies where we can hunt for the very specific targets, and these targets will give rise to very specific medications. You know, there is certain medications which are very good in their nature, very good and very effective in many of the disease. For example, for a beautiful example is turmeric, curcumin. So curcumin is the active component of turmeric. If you start searching, you will find like millions of the papers on the turmeric. So a huge amount of research has already been done on the turmeric. You'll find many papers in the Nature, Science, everywhere. But you won't find a single FDA approved medicine of turmeric, of curcumin. So you won't find any drug for cancer or for any inflammation with the curcumin. The reason behind that, it alters everything. It affects AP1, it affects NF-kappa B, it affects retinoblast, everything it get altered. It affects the inflammatory markers, and that is the reason that it is not having a specific target. So a primary requirement of being a drug or getting developed into a drug is that it should be very specific, and that's why having a specific target is a vital task. So here comes the translational and genetics to understand this specific target and this specific molecule for that purpose, and that has actually completely transformed the drug discovery process for the oncology and rare diseases. And regarding the drug safety, you know that we have the various testing platforms. I'll be discussing about the various zebrafishes and then C. elegans and Drosophila, that how we can design or we can find the drugs for even orphan drugs. Like you can individualize the therapy. You can take the gene of one individual, transform it to any of this model organism, and you can find the specific things. So this targeted validation, and this has affect the cost up to a huge, because it has reduced down to from millions to billions of the cost of the overall drug discovery process. And you know, that's why the gene targets become very important. When we go for the gene targets, there is important to understand the mechanisms, then pleiotropic and generalized versus undesirable effects, druggability, chemical tractability, prediction of drug effect size for the lifelong exposure, that is related to genetics, position in pathways, tissue specificity, deliverability, etc. So these all are the questions which can be answered with the specific target identification and validation process. So these are the various ways how we can find the very specific way of drug discovery process. So I already told you that the genetics and the complexity is increasing complexity leads to the latest and various types of the approaches for the drug discovery process. So I told you that the drug discovery process has concentrated now to individual, and that's why whatever the new drug discovery is happening, it is very focused for the individual and is having very specific tools to understand that what are the various traits which need to be defined, what are the specific diagnostic and prognostic markers for the specific type of diseases. So they are looking more into the specificity in the individual. So when we talk about the various strategies, here comes the few names like the 3D cell culture based approaches, novel applications in RNA interference screening, small interfering RNAs screening, gene editing for the drug discovery process, CRISPR technology, which is actually completely transform the process. So once it is launched, it will completely transform the whole disease treatment way. There is a number of novel platforms for the gene-based drug discovery process, DNA coding, library screening. So these are the various way how the things has been transformed. We'll see it one by one that how things are happening in this drug discovery process. You might have observed that there is various way of culturing the cell for cancer or for many other toxicological studies. We take the actual human cells, we culture in the flask, we aid with the drugs and try to observe the change in the expression of the genes and how these cells are behaving. The very important issue is that when we culture the media, culture the cell in the media, this is having some cells around this, but this is not having the actual environment which they are having in their body. So in respective bodies, there is a complete environment of around the cell which affects its growth and development, its mutations, its a various expression process. So the chemical expression and the outward products are also happens because of the nearby environment of the cell. So cell milieu plays a very important role in its ultimate action. So when we are using culture media, it is surrounded by the cells only and the specific media only. In that, it cannot exactly function as a cell which is functioning in the human body or animal body. And that's why it cannot be useful or as conclusive as the cells in human body. And that's why we cannot replace in vivo models. So here the 3D cell culture is completely transformed this, and it is actually achieving a great success rate. You can see that 900 anti-cancer therapies evolved, but only 12 could find the way from the FDA. And it included, it required the investment of billions of dollars, but still it has not qualified for the approval. The reason is this, that what model we are using and how robust, how effective, or how extrapolable it to humans. And that's why the 3D cell culture has evolved out. So 2D is not useful in many reasons because there is extracellular matrix component which is absent. Cell to cell contact is there only. Cell to matrix interaction is also absent. So there is a requirement of the replication of the same environment as in vivo. And that's why there is a very important role of 3D cell culture where we are using the things in that way. Look at this tumor which is present in the body. So a tumor is having various zones. So outer zone is the proliferative zone, which is having even a supply of the blood. And this has good perfusion of blood. It has interstitial pressure relatively less because there is a good perfusion. Oxygen supply is good. Extracellular pH is same as the blood pH. Proliferation is good, and drug delivery is also possible in the outer area because blood circulation is good. Just compare with the central core of the tumor. This tumor is having necrotized tissue, so there is no proper perfusion because blood vessels are not perforating into it. Interstitial pressure is very high because all the blood vessels, things are going here, is blocked. There is almost no oxygen, so it is a zone of hypoxia. The extracellular pH is widely varied. Proliferation is not there, but it may be detrimental and may be releasing the toxins. And drug delivery, we cannot assure. So such type of the tumors, we cannot touch or we cannot treat the inside areas. And even when we testing the drug in the normal cell culture, we find the drug is very effective. But when we go at the in vivo level, the drug may be effective for the outer zone, but it won't be effective for the inner zone area. And that's how the drug will finally become ineffective after the killing of the outer zone area. And that's why drugs could not find the way out from the FDA. Okay, so this is the things which can be altered or which can be handled by the technologies like 3D cell culture. So there is a quite difference in the various parameters like morphology, proliferation, and stages of cell cycle, exposure to the medium and drug. gene and protein expression in the various conditions like 2D condition and 3D condition, and drug sensitivity analysis in 2D and 3D conditions. So that has completely transformed, and 3D cell culture is actually behaving like in vivo studies. And that's why we need to put up various strategies which can improve this. And this has evolved out with the advent of various matrices, which are actually synthetic as well as the natural, to actually mimic the cell signaling pathways and cell communication things. So that can be present in such type of the models. So this model will be having all the sort of actual things which is actually going on in vivo models. So drug screening is quite more effective and more outcome based when we are using 2D cell culture compared to the 3D cell culture. Of course, there is certain limitations with this also, and this includes the absence of vasculature. There may be size vary in the size of the model. It may be expensive, and there may be variation in the biological derived matrices, because this matrices will be synthetic. It may be of various polymers and various types of the material, but it may not be the actual one which is present in vivo. So these are the few limitations, but still the outcome is far better than the conventional methods. So there is various scaffolds which are present in market in current time. Scaffolds made up of polymeric hard scaffolds, biological scaffolds, and micro pattern scaffolds. There may be non-scaffold based systems like hanging drop microplates or spheroid microplates, which may be having ultra low attachment coating. Microfluidic 3D cell cultures are there, which are microfluidic is really transforming the drug discovery process. I'll show you that how it is happening. These are the various scaffolds which are available, and we can put the cells over this, and they actually function, or they actually looks like or function like it is the organism. In fact, there has been some experiments done with this by coating this with the liver cells and kidney cells, and it has been tried to create the artificial liver and artificial kidney. So that is a different way out in the designing the organs with the similar type of scaffolds. So this can be engineered as a bone, cartilages, ligaments, skin, vascular and neural and skeletal muscle tissue. So we can have such tissue outside the body in ample amount, so that we can have the exact environment as we have in our body. So these are precisely more accurate, having accurate results, and the chances of rejection or the chances of having the results which are not up to the mark is relatively quite less. There is biological scaffold also, which has been designed with the fibronectin, collagen, laminin, gelatin, etc., and they are having almost same gene and protein expression profile as of the in vivo things. So that has also been emerged out. There is number of surface plates which are micro pattern plates which are present, which are actually mimicking like the organs after the mounting of the cells. So you can see that these are the various cells, and they are having various roots here. So this may act as the liver microvasculature, from where the blood may be flowing, and whatever the enzyme they are releasing, that enzymes can be working as metabolic enzymes to metabolize the things which is present in the blood, which is flowing through this. So there's a microscopic structure. So such type of structure can be manufactured now, and that can mimic the actual organ system. So this is the agitation-based approaches of keeping, like this is a hanging drop method, which is actually giving the actual milieu of the environment where the cell suspension can be used for the testing of the drug. These are non-scaffold based 3D cell culture. So you can see that there is, I'll show you here, microplate. These are the ultra low attachment coatings. So the certain coating is actually already done, which is pre-filled, and based on that, the actual environment which is present in the human body is actually mimicked. This is microfluidic 3D cell culture. So these are the various reservoirs, and this reservoirs contains the various chemicals or which are having a composition like the lymph or maybe a specific fluid in the specific body like CSF or blood, and we can control the amount of this which is actually reaching to the cells. So we can even alter the environment also. For example, if you want to see activity of a drug in the vaginal fluid and in the vaginal area or in the uterus, so there is always a changing of the environment of uterus and vagina in coordination with the menstrual cycle. So based on that specific environment, we can alter the environment of these wells, and we can find very specific application in that particular area. So we can find out, even we can mimic even a very complex type of physiological aspects of the human body outside the human body in this type of the synthetic materials. So in that way, we can find various proteins, various gene expression profiles, and various other biomolecules in their native form, which is actually present in the human body. So such type of technologies are in the process, and that's why the success rate is relatively higher. This is RNA interference screening. There is a small RNA, microRNA. Small RNA is around 30 base pair long. MicroRNA is around 6 to 8 base pair long. So this actually regulates the expression of gene in variety of eukaryotic organisms. So I'll just show you a small video so that you can understand that how this small and microRNA can do the gene editing processes. So, is it audible, sir? I hope it is audible.
Scientists have been making rapid progress in understanding RNA interference, or RNAi. Many organisms use RNAi to control genes, and it can also be used as a tool in the laboratory, and in the future perhaps as a therapy. This animation will introduce you to the principles of RNAi involving two important types of RNA molecule, small interfering RNAs and microRNAs. Eukaryotic cells have many sophisticated ways of controlling gene expression. In the complex environment of a cell, these mechanisms need to be precisely targeted. There's a group of mechanisms that use small RNA molecules to direct gene silencing. This is called RNAi. Inside the nucleus, most genes that encode proteins are transcribed by RNA polymerase II. The primary RNA transcript is processed by splicing and forms a mature messenger RNA, sometimes called mRNA. The messenger RNA is then exported from the nucleus into the cytoplasm. Here, ribosomes catalyze translation of the messenger RNA to form polypeptide chains that fold into proteins. But this is also where some small RNA molecules can have their silencing effects. There are several types of regulatory small RNA. Small interfering RNAs, known as siRNAs, are derived from longer double-stranded RNAs that are either produced in the cell itself or are delivered into cells experimentally. The introduction of siRNAs, or double-stranded RNA, is widely used to manipulate gene expression. MicroRNAs are another type of small RNA. Most microRNAs come from RNAs that are transcribed in the nucleus, which then fold and are processed before being exported into the cytoplasm as double-stranded precursor microRNAs. The double-stranded precursors of microRNAs and siRNAs bind to dicer, which is an endonuclease protein that cuts the RNA into short segments. Most siRNAs and microRNAs are approximately 21 nucleotides long. The short double-stranded RNA then binds an Argonaute protein. One strand of the RNA is selected and remains bound to Argonaute. This is called the guide strand. The combination of the RNA and Argonaute, along with other proteins, is called the RNA-induced silencing complex, or RISC. siRNAs direct RISC to bind to specific messenger RNAs. The targeting is precise because it's determined by base pairing between the siRNA and the target messenger RNA. siRNAs often have perfect complementarity to their target sites. Once bound, Argonaute catalyzes cleavage of the messenger RNA, which will then be degraded. MicroRNAs also guide RISC to messenger RNA.
So I hope you understood that this specific small RNA is binding with a particular area of the gene or particular area of the RNA and cleaving this. So if it cleaves the RNA, then it won't be available for the development of a specific protein. So in that way, it can control the expression of certain proteins, and that's how it can prevent some disease to happen. So with this RNA, we understand that there is some gene which is malfunctioning. If we have complementary RNAs, and then it will go inject it will go in the cell, it will affect the gene products of that specific malfunctioning gene, and then it will cleave. So even after the mutation in that particular gene, the disease will not precipitate. So some person may be having a specific mutated or malfunctioning gene, but still this won't happen. So having that specific RNA is really going into a new direction of finding the diseases target. So once we will be having many such RNA and we'll administer such RNA in the various disease condition, and we'll look into it that which disease inhibited by which RNA. In that way, we could develop this RNA as a drug. So this is the part of drug screening in that way.
Part of the microRNA, known as the seed, pairs with a target messenger RNA. This imprecise matching allows microRNAs to target hundreds of endogenous messenger RNAs. Targeting by a microRNA can lead to messenger RNAs being degraded or translation being inhibited. Argonautes and their small regulatory RNA cofactors are found in plants, animals, fungi, and some bacteria, and they are important in a multitude of biological processes and as tools continues to be revealed.
So these are naturally also present in the human body, and even not on the human body, but also in the fungi and many animals also. So this RNA basically used for the genetic screening, for the screening to identify the specific gene which are relevant for a specific pathway which is involved in the disease development. And in that way, we can affect the structure and function association of the mutant phenotype by gene knockdown process. Okay, so this is how the RNA interference, RNAi, can facilitate the finding the specific gene and their product in the process of the disease, and with that we can find the new treatment. Okay, in chemical screening, RNAi screening is amenable to miniaturization and automation, facilitating the high throughput studies. Okay, so this can be useful in finding the new targets for the existing drugs, and that's what we call it repurposing. So we use the drugs which is already there in market for a different disease by similar way. Like there is many inflammatory drugs, anti-inflammatory drugs, which has been used as an anti-cancer, even anti-diabetic, after finding such type of specific activity against few genes. So there is number of such stable ways of doing this, like lentivirus and then viral vectors, adenovirus, and the transfixion or electroporation method with the plasmid DNA, and so on. So there is many ways how it can be used for this purpose. Then microRNA, as I told you that it has even smaller than the small RNA. It is 6 to 8 base pair, that's all. And how it is doing, affecting the process that we already seen in the video. And the very interesting technology is the CRISPR technology. In this CRISPR technology, there is one guide RNA along with this Cas9. It actually affects a very specific gene area. So in the very specific sequence of the target DNA is actually altered by the whole process, and it completely transforms the malfunction type of gene. So CRISPR is like transforming the things dramatically, especially in the screening process and especially in the treatment aspects also. So soon it will be in market, I hope. This is just one example that the same type of the seed, which is having a very high amount of the leaf and shoot and very few roots, but when we transform the gene product, you can find that there is such an amount of roots are present and there is no shoot at all. So this is how the DNA is actually transformed, and the specific characteristics has been induced to produce a specific part of the anything. So if in this plant the root is of interest, so we can actually cleave the gene by the CRISPR to develop a specific phenotypic characteristics. So just have a look about a very interesting video about the CRISPR, what it is and how it is.
CRISPR is an acronym that stands for, first question is, the first question is, what is CRISPR? CRISPR is an acronym that stands for Clustered Regularly Interspaced Short Palindromic Repeats. CRISPR was originally part of a bacterial defense system, but is now used to quickly edit genes in a host organism's genome. Usually, CRISPR refers to the CRISPR-Cas9 system, which can be used to target specific areas of the genetic code and edit the DNA at these sequences. CRISPR can be used to treat the causes of many diseases. The second question is, how does CRISPR work? There are two key molecules in the CRISPR system. One is the enzyme Cas9. Cas9 is a molecular scissors that can cut the two DNA strands at a specific location in the genome to remove or add pieces of DNA into the genome. The second key molecule is a piece of...
So this is the enzyme which cuts the specific gene and then insert the another gene into it. So in that way, we can manipulate the gene. For example, if we are screening for a particular disease, and if we want to know that which gene is actually causing, which gene is malfunctioning and that is causing the disease, so we can cut the specific portions of the gene, insert the right portion, and we can see if there is disease or not. So in that way, the disease screening can be done by the CRISPR. Not only screening, this can also help in the repair process of the gene.
RNA, otherwise known as guide RNA. This RNA is also called the CRISPR spacer sequences, and it is a pre-designed RNA sequence that is around 20 base pairs within a larger RNA scaffold. This scaffold binds to the DNA as a marker for the Cas9 for where to cut, and this ensures that the Cas9 cuts at the right part of the organism's genome. This guide RNA has RNA bases that are complementary to those of the target DNA sequence in the genome, and therefore is able to cut at a specific sequence. So after the DNA is cut out, the cell recognizes that the DNA is damaged and tries to repair it. At this point, scientists can introduce new genes into the genome of the cell. The third question is, where did scientists find CRISPR in nature? A similar system is found in bacteria that is used to respond to pathogens and viruses. The bacteria use this system to snip out parts of pathogenic DNA and store it in their bacterial DNA, to recognize the pathogen the next time it attacks. This is
So
analogous.
this is like immune process of the bacteria, that they can understand that who is attacking this itself, and they keep the part of gene to identify that pathogen.
Wanted posters of the pathogen in its bacterial DNA. Use is similar to a primitive bacterial immune system. The fourth question is, what can we do with CRISPR? CRISPR can be used to target and replace genes in organisms such as monkeys, rabbits, mice, pigs, and many more. In the future, it can be applied to humans to replace mutated genes such as the genes that cause diabetes and other diseases. CRISPR can also be used to induce large deletions or genomic rearrangements, such as chromosomal translocations or inversions. Other applications include transcriptional regulation and epigenetic modification at specific gene loci. For a future outlook, CRISPR holds a large potential as a tool to treat a range of medical conditions that are based in genetics, including some types of cancer, hepatitis B, or high cholesterol. Another possibility includes the editing of genomes of germline or reproductive cells in humans. However, this may not be applied anytime soon, since there are many ethical considerations, such changes to genomes in germline or reproductive cells are heritable or passed down through generations. The final question is, why is CRISPR so revolutionary? There are many other types of genome editing tools that biologists can use. However, CRISPR is revolutionary since it can be used to target any gene in a target organism's genome. The only change that is required is in the crRNA or guide RNA to change the targeted gene. Therefore, CRISPR is really efficient and customizable compared to many other gene editing tools. For instance, other gene targeting and editing tools like zinc finger and talons or transcription activator like effector nucleases require a complete redesign of the protein DNA interface to change a target gene. Additionally, all the possible locations for cuts by the CRISPR-Cas9 complex can be easily kept in a genomic library of guide RNAs, making it really easy and simple to choose which genes to cut out. Thanks for watching. Please leave a like and subscribe.
So this is really a revolutionary way how the gene editing can be done, and many of the scientists who are actually into it will be understanding that it is really a great concept to edit the gene whatever they actually want to. Okay, so in that way, it is really very useful. A very interesting aspect is with model organisms which has evolved recently because of the various genomic and proteomic things which has evolved on. Technologies which has been evolved. The examples are C. elegans. It's a very small worm. So a very small worm, it's just 1 millimeter worm. It is having a very countable cells, like somewhere around 782 cells in its body. It has a very specific number of cells for the kidney, nervous system, GIT, brain, and all these specific organs. So if you want to taste, and moreover, it is a transparent one, and you can culture that into a petri dish. So you need not to have a very big or very expensive setup for culturing the C. elegans. So such a small, such a transparent, where we can transform the gene and we can see any fluorescent or any sort of gene to see in microscope also. So this is really a revolutionary type of organism where we can insert a gene and then we can see the gene products and their toxicity on the specific cells of the body, and that we can extrapolate over